As I have discussed in the lecture also, we are going to. Am I audible to everyone?
Yes, doctor.
Yes, doctor, we can hear you.
So I am going to talk about the important thing, but it doesn't mean that whatever I did not touch or I did not say is not at all important, that it is not going to come in the exam. It may come, but whatever I am going to point out, the thing are more important, and there are more chances of coming in the exam. So it is better to read everything, but more on this, whatever I am telling you. In addition to that, please do not ask about that thing is not important, what you have not said, because I cannot say that, because that thing can also come in the exam. I have already mentioned this thing in the male side lecture, so I just want to communicate if there is a female student, they have not got this information. Now let's talk about what are the things that are important and can give us more edge in getting the good grades in the exam. So, Bismillahirrahmanirrahim. This just remember that all the amino acid in our body is having L-alpha configuration. And all amino acids are chiral, that is asymmetric except glycine. All amino acids are amphoteric, that is they are having acids as well as basic property, and they can be cation, anion, and the zwitterion depending upon the pH. So they can be buffers because of this property, and histidine is having the maximum buffering capacity because of the presence of the imidazole group. And protein is the major buffering system in our body, if we are talking about the entire body. Concerning the PI, you have to, PI is pH. If the question comes regarding the PI, remember that it is a pH actually. Like we have discussed in the Km. Km is the substrate concentration at which half of the maximum velocity attained by the reaction. Exactly in the same manner, isoelectric point is basically a pH at which the molecule exists as a zwitterion. So it is a pH. Now coming to the classification. All the classifications are important. Okay. Some are more important, like branch chain amino acid. The acidic is basically aspartate and the glutamate, and the basic are arginine, histidine, and lysine. These are more important. This is very important, polar and non-polar. What are the polar and what are non-polars? This is important. I think we have asked one question in the quiz also concerning the utility of this. The methionine is a methyl donor, is major methyl donor in the reactions. The abnormality in the sickle cell anemia, that is the glutamate is being replaced by the valine. Excuse me, guys, try to stop your mic.
Doctor, I'm sorry. Can you repeat what you say?
Actually, the sickle cell anemia, in which the glutamate is being replaced by valine, and the methionine is the major methyl donor as an amino acid in the body. And with this first point we already discussed in the first slide of the classification. And the glycosylation, that is addition of the carbohydrate, happens at 3 amino acid, serine and threonine at O-linked and the asparagine as N-linked. Cysteine is responsible for disulfide linkages, disulfide bond. Now coming to we can skip the nitrogen balance this time. Inshallah, in the final we will discuss. Nutritional essentiality. We are skipping this also for the exam now. Inshallah, this is very important. Glucogenic, neogenic, and the ketogenic. Very important. So as I told you how to memorize, you just have to memorize these two. The rest of the thing, because it is an MCQ, multiple choice question, so you can answer easily if you have memorized these 6. Okay, 4 in the both gluconeogenic as well as ketogenic, and 2 in the ketogenic. So remember this very well. Now coming to the derivatives. Derivatives are very important. The melatonin, serotonin, and the niacin is the derivative of tryptophan. Dopamine, norepinephrine, and epinephrine is the derivative of tyrosine. Histamine is for histidine. And nitric oxide is for the arginine, and spermine is for the ornithine, and gamma-aminobutyric acid is for the glutamate. This is all in this session. Now we will go for the another lecture. You have question?
Doctor, can you repeat from the beginning, because we didn't follow.
Dear, you can ask with your friend. Did you get that?
Okay.
Okay. I think your friend will help you. If you want to record this, you can record it. There is no problem. I cannot record it. Okay.
Doctor, what do you say about this?
In this slide, I told you that nitric oxide is a derivative of arginine amino acid. You have to memorize that which derivative is from which amino acid, because usually the direct questions will be there. That which of the following is the derivative of the tryptophan, or in another word, like serotonin is the derivative of which of the following amino acid, or serotonin can be synthesized from which of the following amino acid in our body, like that. It's a simple question, direct. But if you don't know, you cannot answer. Therefore, you have to memorize this table. It's only 6 or 7 items. Now coming to the second lecture, in which we discuss about the protein structure and different kinds of function of the protein. So the different kinds of bond, they are present mentioning, as just go through once what are the different kinds of bond present in the protein. And this peptide bond, the characteristics of the peptide bond, what are peptide bond, and what is the characteristic. The glutathione is very important. We will come to the glutathione in the next slide. Glutathione has got 3 amino acid: glutamate, cysteine, and glycine. They are having 3 amino acid. This is the reason they are called as tripeptide, although they are having two peptide bond only. Okay, the one peptide bond is this, and the another one is this. This one is false peptide bond, and this one is true peptide bond. We already discussed why it is false and why it is true, because the carboxyl group linked with the gamma carbon. This is alpha carbon, this is beta carbon, and this is gamma carbon. So the carboxyl group here is linked with the gamma carbon, whereas the amine group is linked with the alpha carbon here. And the carboxyl group is linked with alpha carbon, and amine group linked is with the alpha carbon here. So it should be both the carboxyl and the amine, the peptide bond should be linked with alpha carbon respectively to their amino acids. However, in this bond, the carboxyl group is attached with the gamma carbon, not the alpha carbon. This is the reason they are false peptide bond, but they are peptide bond. Now, in the next important thing is that the glutathione is required as a detoxificant or the antioxidant, and they are very important to detoxify hydrogen peroxide inside, especially inside the RBCs. And in this process, glutathione itself gets oxidized and NAD pH is required. So remember that NAD pH is required to regenerate the glutathione, regeneration of the glutathione. And this NAD pH continuous supply is being met by glucose 6-phosphate dehydrogenase enzyme. And if there is a deficiency of this enzyme, the NAD pH will not be available or cannot be generated, and glutathione will be decreased or present in majority of the oxidized condition, and that will lead to lysis or breakdown of the RBC membrane. Therefore, the RBC will die after that. You got this or not? Because this is a very important slide in which it is mentioned that glutathione is going to protect the RBCs. If there is a decrease in the glutathione, the RBC membrane will be broken down due to damages mediated by hydrogen peroxide. And for the regeneration of the glutathione, NAD pH is required. NAD pH is being generated through glucose 6-phosphate dehydrogenase enzyme. So if there is any deficiency or any defect in this enzyme, NAD pH quantity will not be produced in the appropriate amount. Now we will proceed, that is the alpha helix. What kind of bond is this structure? Alpha helix, the alpha helix is the most common, and the bond that stabilizes the alpha helix is hydrogen bonding. And the structures that can disrupt the amino acid that can disrupt the alpha helix is either they are small or they're having a different shape, or they are bulky, or they are charged. Now coming to the second kind, they are also being stabilized by hydrogen bonding. And the third kind, that is trans loads or bends, they are being stabilized by hydrogen bondings as well as ionic bonds. Now that we will skip this. Now coming to the tertiary structure, it is a 3-dimensional structure, and it is more important, although it can happen only after the secondary one. So these are the bonds or interactions or forces that can stabilize the structure. And this is very important that it is happening in the endoplasmic reticulum. This slide is very important. They are happening in the endoplasmic reticulum. And majority or some of the proteins can fold spontaneously, but some protein need help, and the help is mainly provided by the molecular chaperones or simply chaperones. In the exam, there are chances only mentioned as chaperones, which are the following proteins is required for the protein folding. So if it is not necessarily mention molecular chaperones, only chaperones, please turn off your mic. Only chaperones may be mentioned in the option, okay? Or maybe molecular chaperones may be mentioned, or maybe heat shock proteins are mentioned, like here, or maybe chaperones are mentioned. So anything can be mentioned in the option. So you have to choose whatever is there. Now the protein denaturation, these are the factors that can denature the protein, and in the denaturation, it is the loss of tertiary structure only. After the loss of tertiary structure, the protein becomes non-functional. Prions are basically misfolded proteins. When they come in contact with the normal protein, they can also misfold the normal proteins in our body, and that can lead to abnormal turnover or the recycling of the proteins in the cell. These are the diseases. Some of these diseases can be in the humans, like Creutzfeldt-Jakob disease, Kuru. These can be in the humans. Quaternary structure can only be present in those proteins that are having more than one polypeptide. In the one polypeptide, there will be no quaternary structure. More than one polypeptide, there can be a quaternary structure. I know that you don't know all proteins that is having more than one polypeptide. So I will ask only those proteins that are mentioned in these slides, whether they can have a quaternary structure or not. We are skipping this slide for now. Maybe this also, but this slide is important, conjugated protein. What are the different kinds of biomolecules that can be present in combination with protein, like carbohydrate and the phosphate, like lipids and nucleoprotein? They're examples. We are skipping this also because gradually we will talk about this in different module. Now coming to the buffers. In the buffers,
Doctor, I want to check myoglobin is secondary or structures.
myoglobin has got till tertiary. There is no quaternary structure in myoglobin because they have got only one uh, polypeptide. Now coming to the, skip the acids and bases. These are very basic things. The in the hospital we used to measure pH using the arterial blood gas analyzers, and that uses the ISE technology, that is ion selective electrodes or electrolyte analyzers. We are skipping these slides. The buffer, the definition, just go through the definition at least once, so that if the question comes, you should not be. Yes, the composition of buffers. This is very important slide. The how we call anything as a buffer. When we call it, when they are having weak acid and their corresponding salt. This is very important. Two things must be there to make it a buffer, or weak bases and their salts. Okay. So just get the definition is very important, along with some of the examples. Like the examples are easier to memorize because it will be a multiple choice question. You just have to solve whether that particular distractor has the combination or not, that is acid and the salt. And the way to see it, like here, more number of hydrogen is usually an acid because it is going to donate a hydrogen in the solution. So, and the upper part that is mentioned here is the salt because it is having less number of hydrogen. The other thing is that whether it is extracellular or extracellular has got sodium in them, in the salts, and the intercellular has got potassium in their salts. So this is not very difficult, and there are two things. Acid can be identified by more number of hydrogen. The salt can be identified by the presence of cations. Whether it is extracellular or intercellular, it can be identified by the presence of sodium or potassium. If it is sodium, it is extracellular. If it is potassium, it is intercellular. Erythrocyte has got a specific kind of protein, more, that is hemoglobin, so you can have an idea. Rest of the thing, as you can see, these both are same. This and these are same. So both are same. The only difference in the protein, here there are different kinds of protein, but here only hemoglobin mainly in the RBCs. Now coming to the next slide, how the buffer works. This is not, we are not going to ask, although you should know, mainly in the writing it is needed, but for the MCQ, we don't ask. The different kinds of buffers, that is what is included in the bicarbonate buffers, what is included in phosphate, and what is included in the protein. Hemoglobin is the same, actually it is also a protein, but it is present especially. Now if the question comes, which of the following buffer system is the major buffer system in our body, then the best answer is the protein buffer system, if it is present in the option. If it is not present, then you can check the bicarbonate buffer system. But if the question, it depends on the question. If the question asks which of the following buffer system is the major buffer system in the plasma or in the blood, then bicarbonate buffer system is the best answer. Even if the protein buffer system is present, it is not the best answer. You got the concept or not? So pay attention to the questions. And as I mentioned, for the calculation of the pH of the system, you need to have three things, and that I think you have already practiced that at your home, and in the quiz also there was a question. So I think it will not be difficult actually. There are only three things you need to calculate. If you remember, if you memorize the equation, Hasselbalch equation, then you can do it. So that is pH is equal to pKa plus log base over log acid. All the phosphate buffer system are also important. Now coming to the protein buffer system, at least you should know what is the part and what is the salt part. This slide is very, very important. That is hemoglobin buffer system. As I told you that there are many groups in the hemoglobin, but which of these group is mainly responsible for the buffering capacity of the hemoglobin? It is the histidine amount. So if all these options are present, and the imidazole group is the best option to choose in the exam. Now we are going to the third session, that is the enzyme. Remember the allosteric site, apoenzyme, coenzyme, and holoenzyme. What are these? Classes, as I told you in the classes, you have to memorize the group and the example only. So the questions will be very straightforward. That is, which of the following enzyme belongs to the group of oxidoreductases? Then you have to choose if the option contains lactate dehydrogenase, you have to choose it. Or in other words, the question may be like lactate dehydrogenase belongs to which of the following group of the enzyme? If the option contains oxidoreductases, you have to choose it. So the same thing, the transferases and the serine hydroxymethyl transferase or the amino transferases. The category and their example. Category and their example. Lyases, pyruvate decarboxylase, isomerases, and methylmalonyl CoA mutase, ligases, and pyruvate carboxylase. So these 6 different categories, their name and what is the example of the respective group. There are 3 factors that can influence the activity of all kind of enzymes, and in this we discussed about the substrate concentration, that at certain substrate concentration, the rate of the reaction is half of its maximum, and that particular substrate concentration is known as Km. We talk about the Km, that is Km means if high Km means low affinity of the substrate with the enzyme, and low Km means high affinity of the substrate with the enzyme. Just go through this equation also. We need 3 things to calculate the equation. It is not very difficult. You can calculate if you practice a little at home. This is important actually, very, very important. The significance of the Km, importance of the Km, what we understand by the Km. Km is basically the concentration of substrate at which the rate of the reaction is half of its maximum. And if the Km is low, it tells us that the enzyme has high affinity with the substrate. If the Km is high, it tells us that the enzyme has low affinity with the substrate. At temperature, we have an optimum temperature. I think we already asked this question in the quiz. And the pH, every enzyme has their optimum pH, their own optimum pH. It depends what kind of enzyme, whether they can work best in the acidic medium or in the alkaline medium or in the neutral pH. So this is all about the buffers, enzyme 1. Now we are going to have the enzyme 2. In the enzyme 2, there are inhibition and regulation, both we discussed. There are 2 types of inhibition, irreversible and reversible. There are examples for them. The reversible is of again two types: competitive and non-competitive. Okay. These are the example of why it is flipped. Anyway, the aspirin, penicillin, and 5-fluorouracil are the example of the irreversible inhibitors. Cyanide and fluoride are also irreversible inhibitors. But these three, the bottom three, are used as drugs. One of them is used in the diagnostics, and the other one, lastly, is used mainly as in the research for the analysis. Now coming to the reversible inhibition, that is competitive inhibition. Remember this point, the last point, these 2 points. That is increase in K and there is no change in Vmax. So if you want to save time, apart from the examples, there is a slide that mentions both of these reversible inhibition together, in which it is mentioned that substrate binding site is the site where the inhibitor binds in the competitive inhibition, and allosteric site is the site where the inhibitor binds in the non-competitive. There is an increase in Km in the competitive inhibition, but there is no change in Km. And there is no change in Vmax in the competitive inhibition, but there is a decrease in the Vmax in the non-competitive. So memorize this very well, because this is very important. Well, what kind of changes are there? Just memorize in competitive inhibition what happens, in non-competitive what happens. Now coming to the examples. Examples are very important. That is sulfanilamide can inhibit the para-aminobenzoic acid synthesis of the folic acid by competitively inhibiting. It is a competitive inhibition. It is a competitive inhibitor. So the question may be in the exam that how the sulfanilamide inhibit the synthesis of the folic acid. It is through the competitive inhibition. And exactly in the same manner, the statins also inhibit using the competitive inhibition because of the similar structure as the substrate. Now coming to the regulation. In the regulation, there are rapid regulation and the slow regulation. In the rapid, there is allosteric regulation, proteolytic cleavage, and covalent modification. In the slow, it is induction and reparation. Okay. So allosteric regulation is very important. We have talked about, although it is not very obvious here, but it is, if you see in your own slides, which is mentioned that homotropic is when the substrate itself binds at any other site, that is allosteric site, and can influence the activity of the enzyme. We call it a homotropic influencer or effectors. And feedback inhibition is a type of allosteric regulation. Feedback inhibition. The covalent modification is phosphorylation and dephosphorylation. Phosphorylation is being done by the kinase enzyme, and dephosphorylation is being done by the phosphatase enzyme. Some of the enzyme produce a zymogen, and they can be activated after removal of some of the peptides or some of the amino acid. Induction means increase, and repression means decrease in the synthesis of the enzyme. Now we are coming to the last topic, that is lipid chemistry. So do not take anything lightly, please. What are lipids? We are past that. Functions, we are just skipping. This is important. Classification. The simple lipids. What are simple lipids? That is triacylglycerol, simply fats. They are made up of 3 fatty acids and a glycerol. Exactly in the same manner, waxes are basically esterified with long chain fatty acids, long chain alcohol. Fatty acid is esterified, like for example cholesterol esters. Now coming to the compound lipids here. This is very, very important slide. That is dipalmitoylestine. This phospholipid is basically responsible for the uh, preventing of collapse of the lung during the respiration, and it acts as a lung surfactant. So in case of deficiency due to the premature babies, the premature babies are prone for having respiratory distress syndrome. So we have to treat them with synthetic form of the made in the industry, the dipalmitoylestine. So what is exactly dipalmitoylestine? It is the two palmitic acid attached with a phosphate and choline to a glycerol. You got the point? The question may come in many ways that which of the following phospholipid is which of deficiency of which of the following phospholipid may lead to respiratory distress syndrome in premature babies? The answer is the dipalmitoylestine or the dipalmityl phosphatidylcholine. Or the question may be like dipalmityl phosphatidylcholine is made up of which of the following sets of the molecule? The answer is glycerol, 2 palmitic acid, 1 phosphate, and 1 choline. So either way, the question may be. And the exactly same, the rest of the thing will be easier because if you know this is the phosphatidic acid, along with any ethylamine or the serine, these are basically phospholipids that are mainly present in the plasma membrane. Now coming to the sphingomyelin, what is the component of sphingomyelin? Sphingosine plus fatty acid plus phosphate group plus choline. Okay. So in the exam, the question may be, this is very important slide, very, very important. The question may be like that, which of the following sets of molecule makes a sphingomyelin? And the option may be sphingosine, fatty acid, phosphate group, and choline. You have to make sure you choose the best answer because if the option mentions 2 fatty acid or 1 fatty acid or 1 phosphate group, 2 phosphate group, choline or ethylamine, you have to choose the best one. What is mentioned here? Like sphingosine, 1 sphingosine, 1 fatty acid, 1 phosphate group, and 1 choline. These 4 things must be there to choose as a best answer. You got the point? So without, and the ceramide is just the fatty acid and the sphingosine only, without any phosphate and choline. Functions of phospholipids, we already know, just go through these ones. And we are coming to the amphipathic lipid bilayer, glycolipids. We can skip glycolipids now because it will be too much. And now the derived lipids, in the derived lipids, we will mainly talk about the polyunsaturated fatty acids and the essential fatty acid. Essential fatty acid, we have discussed there are only 2 essential fatty acids. Arachidonic acid is conditionally essential. Okay. And what are the things that can be synthesized from cholesterol is the vitamin D, bile acids, steroid hormones, that is sex hormones, glucocorticoids, mineralocorticoids. These are the things that can be synthesized from cholesterol. This is all. Any questions?
Thank you, Doctor.
Thank you, Doctor.
Thanks, welcome. I wish you all best luck for the exam. Inshallah, you will all perform well. If you go through the important things and you go through other things also, but more focus on the important things, inshallah, you will get A+, inshallah.
Thank you, Doctor. Allah bless you, inshallah.
Inshallah. Thank you very much. You're welcome. And have a nice day. Assalamu alaikum.